All right, hi class. So I'm gonna split this into two different lectures. So one will be finishing off the cardiovascular anatomy and physiology, and the other one will be introducing the nervous system. So we'll cover the eye and the ear next week, and then we'll go into computer modeling. So let's just finish this off. So we're gonna backtrack just a little bit in this lecture just to review some stuff. Alright, so just having some, here we go. Okay, so an overview of the circulatory system. So we already saw this slide here in class, but I just kind of wanted to go over it once more. So let's start at the left atrium here where my clicker is, and I'm sorry, the left ventricle, and this is the workhorse of the heart. So this one pumps blood to the aorta, which then gets split off and it goes to the head, but also to the systemic organs, so like the kidneys, the liver, basically all of the blood vessels that are in our extremities. And then they get smaller and smaller and turn into what are called arterioles. Um, and then they turn into what are called capillaries. And capillaries are, hundreds of millions of blood vessels that are sitting in our extremities and in each part of our organs. And that's where basically oxygen is transported across the really thin walls of those capillaries. So then they turn into veins or venules, and they get bigger and bigger and bigger and turn into veins. And they all kind of come back in two major veins, I should say, the superior vena cava and the inferior vena cava, which both drain to the right atrium, and then that drains into the right ventricle, and then that pumps oxygenless blood, or deoxygenated, I should say, to the pulmonary artery, which is the main artery that goes to the lungs. And again, the arteries get much smaller, turn into arterioles, and turn into capillaries, and those capillary beds are the ones that are basically hugging the alveoli that are in your lungs, and then that's where they basically get oxygenation back. And so you can kind of see, like, we talked about the lungs earlier. This is kind of where they come together with the circulation to oxygenate blood and deoxygenate blood. So that's kind of the cycle. Then this blood that's oxygenated comes back to the left atrium, and the cycle kind of continues, and it goes in this circle. All right, and here are some key points that I want you to remember. Blood vessels originating from the heart get progressively smaller. That's gonna lead to increased resistance within each vessel, and we'll see how we model that next time. The total cross-sectional area of all of the blood vessels is the greatest in the capillary beds. In fact, it's almost 500 times more than the biggest artery that you have in your body. And we said last time that's because you have hundreds of millions of them in your body, and so even though each individual one has a small cross-sectional area, altogether they make up more than the area of one big vessel. The heart, the ventricles contract, which causes pulsatile flow. So if we start at the left ventricle, and you were to kind of measure the blood pressure here, it would oscillate up and down, up and down, up and down, hitting a peak and then kind of going down and hitting some minimum, which we'll get to what that value is later. But as you progressively get away from the point of contraction, you'll start seeing that those pulsations kind of die out. And they die out for a couple of reasons. One of the reasons is that, like I mentioned earlier, that the blood vessels are elastic, so they'll store some of that pressure or volume in the expansion of the vessel, and also because you're getting progressively smaller as you go from ventricle to the organs, you're also getting just the dampening of the higher resistance of the blood vessels is what's causing the pulsations to get smaller. That's relevant because when we talk about like measuring clinical like parameters like pressure, flow, you're gonna get different pressure and flow waveforms in different parts of the circulation. So like if you're measuring right in the aorta, right near where the ventricle's opening, you might get some pressure that is closer to what the ventricle pressure is. Whereas if you get much further away from it, like on your fingertip, that will be a lot smaller than what you can measure at the ventricle. And then also conversely as well, on the right side of the heart, you'll measure much different values of pressures. You'll still have pulsations, but they're not going to be as large. So, just kind of giving you a prelude to what's coming later on, specifically with modeling. So where is your heart? We mentioned that if you're looking at your midline, it's a little off center, a little bit to the left actually. So the apex, which is actually the bottom here of the heart, is located on your left side, and it's basically supported, or not supported, but it's on top of the diaphragm, which basically is this structure that separates the thoracic cavity from the abdominal cavity. It's The heart is within this thing called the pericardium, which is the sac that covers the heart, and like I mentioned last time, it may or may not have some function in helping the heart contract. Some of the big arteries I do want you to know are what are called the great arteries, and they include the superior vena cava. So that is the vein that returns deoxygenated blood. I always get this back to the right atrium, and you have the okay. Sorry for that. There's my computer seems to have just died. Uh, just bear with me here. All right, so the superior vena cava carries deoxygenated blood back to the right atrium. What you can't see here is another blood vessel that's, you can call it blue if you want, that's coming up into the right atrium as well. So you have mixing. So this basically brings the deoxygenated blood from the upper extremities, so like your head and your neck area, back to the right atrium. You have what's called the inferior vena cava, which is a large vein that brings deoxygenated blood from the organs in your lower extremities, and that mixes in the right atrium. This is called the right atrial appendage. It's not actually the right atrium, but it's like this piece of meat that's on the right atrium. And then it drains both of them. So there's a superior and inferior vena cava. The other two great arteries are the aorta. So the aorta, again, the heart is not open in this case. You're seeing the outer kind of surface of it. The aorta takes the blood from the left ventricle, and then it, separates into the other arteries, and going to your upper extremities, and even down to your lower extremities. And that's the main one that takes oxygenated blood to your organs. You also have this one here called the pulmonary trunk. That one takes deoxygenated blood from the right ventricle and pumps it to the lungs. And you can see that it's very short distance, so from right ventricle to the pulmonary trunk and the left and right pulmonary arteries, and then it takes that blood to the right lungs to get oxygenated. So that's kind of what you should know in terms of like the big blood vessels in your heart. Heart tissue. So heart tissue is made up of three different types. The most important type, I will say, is myocardium, because that is basically the major muscle cell that's responsible for contraction, specifically in the ventricles. So these myocardium are very electrically susceptible to being stimulated. So what that means is like when there's some change in the membrane voltage across these cells, they're able to basically contract, and that makes them pretty special. On the interior side of your heart, so this is like within the ventricles, you'll have the endocardium. This is tissue that's very smooth, and that's pretty important for having blood flow or blood touching that surface, because you don't want anything sticking to that surface. So that's usually really smooth. And on the other end, you have the epicardium, which is attached to your pericardium. So those are the three that are sandwiched in there. Again, the most important one is the myocardium, because that one's going to be the one that easily contracts. Okay, so this is a view of if you were to cut the heart in such a way that you can see the inside. So if you cut it, so you open it up here. So this is actually an anterior view, so the front view. Let's go through one by one. So there's a couple of things here. And unfortunately it's not colored. I should maybe change this to a colored picture. But let's start with, I guess where they start here, number one. So number one here is the right atrium. And I mentioned before, this is the chamber that receives deoxygenated blood from your lower and upper extremities. From the upper extremities, the blood comes from the superior vena cava. From the lower extremities, you have the inferior vena cava. That all goes into this chamber, and then you're going to hit this structure called the tricuspid valve. So this valve here, it actually has three leaflets. That's why it's called tricuspid. It opens up, so whenever the pressure in the right atrium is larger than in the right ventricle, then this acts like a diode, right? Basically, it opens up when the pressure at the inlet or at one portion of this structure is greater than the other portion. So that pressure gradient causes it to open, and that causes flow to basically drain from the right atrium to the right ventricle. Once that volume gets into the right ventricle, that pressure will build up, and then it'll cause the pressure difference or gradient to reverse. So this will now be at a larger pressure than in the right atrium. That causes this valve to close. Now, what keeps the valve from basically inverting itself, right? I mean, these structures in the valve are very, they're very thin. They're kind of like flaps. And so what causes it to basically reverse back, like when this pressure builds up and gets higher than this pressure in the right atrium, is you have these thing called chordae tendineae. They're basically cords or tendons that keep the valve leaflets in tension and keep them from collapsing into the right atrium. There are some diseases that you can get or you're born with that make these very stiff or make these very easily damaged, that can cause this valve to basically collapse back into the right atrium, which then causes regurgitation of flow going from right atrium to right ventricle and vice versa. So it's very important that these things stay in tension to keep them from collapsing back. So once the right ventricle pressure increases, then you're going to also get some contraction from the right ventricle, and we'll get to where that comes from later. That will then contract, and then the pressure difference between right ventricle and the pulmonary artery is what matters. So when this pressure gets bigger than the pressure in the pulmonary arteries, this pulmonary valve will open and cause flow to come out into the pulmonary trunk, and then it exits to the lungs. When this ventricle starts to basically empty out, the pressure will decrease compared to the pulmonary trunk, and then this valve will close, and you'll still have continuous flow of blood going to the lungs because of the compliance or the elasticity of the artery here. So that's how the right side works. So you have basically two valves you got to keep track of: tricuspid, which connects the right atrium to the right ventricle, and the pulmonary, which connects the pulmonary artery to the right ventricle. Alright, so then moving away then into the lungs, you come back. Once the blood gets oxygenated, it comes back to the right ventricle. So they go to the pulmonary arteries, and then they return back to the left atrium here, number 7. And so you'll have from the pulmonary veins, you'll have drainage from the left and right lungs into the left atrium. And again, this chamber collects the blood. In this case now it's oxygenated blood And then that's gonna, when the pressure in the left atrium exceeds the pressure in the left ventricle, you're gonna have the bicuspid valve open. So that designates only two flaps. These also have chordinae, tendonae, and there's also a muscle that helps to contract and open it as well. Then that drains the left atrium to the left ventricle. And again, once this pressure starts to fill up in the left ventricle, if that pressure is greater than the aortic pressure, you're going to have another valve open, which actually you can't see here. It's blocked by the pulmonary trunk, but it's called the aortic valve. That aortic valve will open and empty out the ventricle, and that's where you get the ejection of blood into the organs. Now, there's two sides to the heart, right? There's a left and the right. There's a left and the right here. They are separated by what's called a septum. That's the piece of muscle that separates these two chambers from each other. Now, this is a really important piece of meat because it basically keeps oxygenated blood that's in the left ventricle and deoxygenated blood in the right ventricle separate. So some babies, it actually turns out, so this is actually a remarkable kind of fact when we're all born. When we're all born, our heart does not look like this exactly. There actually is this small blood vessel, it's much bigger when we're smaller, that does connect the aorta to the pulmonary trunk. That's kind of an interesting thing because this is connecting oxygenated blood to deoxygenated blood here. So you have some mixing of oxygenated and deoxygenated blood. And the reason for that is because when you're a baby in your mother's womb, your lungs are flat, right? So you're not breathing air because you're basically in the womb, and so you're in amniotic fluid. So your lungs are collapsed. So they present a large resistance to your ventricles, or to your right ventricle, I should say. So in order to kind of ease that resistance, you have this artery that connects to the aorta, and then that releases some of the pressure that you need to basically pump to the lungs. So you have a mixing there. What you also have is a hole in your septum when you're a baby that again mixes deoxygenated blood from oxygenated blood. And again, it goes back to the same thing. In order to oxygenate your organs, you have to have that bypassing because this resistance to your lungs is so large. So you're going to have another avenue for oxygenated blood to get to your organs. Okay, so now when you get out of your mother's womb, your lungs start to expand and they become very compliant. So if you're looking at the right ventricle here, this resistance now becomes very, very low. So you're able to pump to the lungs now very easily. What that does is that causes this little connector here to get much and much smaller, or it should at least, and it'll go away eventually. It also causes that hole in your septum to close. Now for some kids that doesn't close up, so they have to have surgery, and that's called an atrial septal defect. So you have your septum goes up to your atrium as well. So that connects the two atrium, and then you have what's even more deadly is the ventricle septal defects, which are basically holes connecting the ventricles. So kind of amazing the transformation that your heart does when you're born, but these structures are within all of us, and they're very important in kind of keeping oxygenated blood and deoxygenated blood separate. Alright, here is a top view of the heart, and you're cutting the heart in such a way that you're able to see the valves. And so let's start over here. So this is they called it the tricuspid valve in the previous slide, but you can also call it the pulmonic valve. So this one here is the one that connects the right atrium. I'm sorry, this is where are we here? Pulmonic, mitral. Got it. Okay, so this is actually the pulmonary valve. They call it. I got confused there. Pulmonic valve. So this one connects the right ventricle to the pulmonary artery. Here, right here you have the aortic valve that connects the left ventricle to the aorta. Here you have the mitral valve, which is basically the bicuspid valve That's the one with two leaflets. That's the one that connects the left atrium to the left ventricle. And then on the right here you have the tricuspid valve, again the three leaves, and that connects the left atrium to the left. I'm sorry, the right atrium to the right ventricle. All right. So that's basically how they look. So you can kind of see the difference. So this one like is if you're like in the if you're in the left atrium and you're looking inward, and when it opens up you'll be able to see the left ventricle. This one here is if it opens up, you're viewing it from the perspective of the right atrium, and once it opens up you'll see the right ventricle. These are the opposite views. So if this is viewing from if you're in the pulmonary artery, and when it opens up you'll see the right ventricle, and then here if they open up you're looking from the perspective of the aorta, and if it opens up you'll see the left ventricle. And this is a colored view of them, an actual meat looking or I should say like an actual heart. So there's basically two phases of the cardiac cycle, and we'll get to this in more detail in the next couple of slides. But I wanted to show you how these valves look when they're in the two separate stages of cardiac contraction. So the first stage, or I guess one stage, is systole. That's when the heart or the ventricles are actively contracting. That means the pressure in the ventricles in the left and right are higher than in the atria, and that also means that the pressure in those ventricles are also higher than in the great arteries connecting to them. So like for instance, if you look at the systole, when the right ventricle the pressure there is greater than in the pulmonary artery, and also greater than the right atrium. In that case, you're going to have the pulmonary valve open up because that pressure difference will dictate that, and you're also going to have the artery from the right atrium to the right ventricle being closed. So this actually shows that. And if you go on the left side, same kind of thing. You'll have the aortic valve open up because of the pressure gradient in the left ventricles pushing flow out, and you'll have the tricuspid valve closed from the left atrium to the left ventricle because of that pressure gradient now reversed. So you can actually see that here in this picture, where both of the what are called the atrioventricular valves, which is basically the valves connecting the atrium and the ventricles closed, but you'll see the other valves open. So like this is the pulmonary valve and the aortic valve. Alright, so during diastole, that's when your heart is actively filling. So what that means is that the ventricles are filling. And in this case, the pressure is higher in the atria than in the ventricles, and the pressure in the great arteries is higher than in the ventricles. So in that case, you're going to have the reverse happen. So look at the schematic over here. The pressure difference from atrium to ventricles are going to be going forward. So you're going to have active flow from atria to ventricle, which causes filling of the ventricles. So you'll have the bicuspid and tricuspid valves open, but conversely, you're going to have the valves connecting to the arteries being closed because the pressure in the arteries are going to be bigger than in the ventricles. So like here, the pulmonary valve would be closed, and also the aortic valve would be closed. So that's shown here, where the pulmonary and aortic are closed, and you have the atrioventricular valves open in this case, filling up the ventricle. And you see here the coordinate tendonae for the valves that are connecting them. So they're very, very thin structures that allow it to open. Alright, so so I like to interject some of the physiology with some actual engineering things here. So an example of, we talked about valves and how they allow for one-way flow, and they're complicated structures that have coordinate tendonae. So one of the first kind of I guess, devices made for the heart are artificial valves. And on the left here, you'll see an image of what an old, or what I should say is how valves have essentially developed over the years. Here are some actual images of them, and then here's an example of what they look like now. So let me go over a few of them. So, sometimes the valves, if you kind of go back, I mentioned before that some of these tendonae, coordinate tendonae, may be damaged or not connected, or you'll have some damage with the leaflets of the valve. There could be all sorts of problems that you can have with valves that essentially don't allow for flow to go in the correct direction during systole or diastole. And the take home message here is that when you have non functional valves, you're going to have a non functioning cardiac cycle, right? And what that leads to is basically you don't get enough oxygen to your organs or to your lungs. And so you have to rectify that. And so one way to do that was to replace the valves entirely. And initially, what you'd have to do is you'd have to have really major surgery to basically disconnect a valve. So let's say that your aortic valve was not working. This is the aorta. This is the aortic valve. Say it wasn't working. So flow, when the left ventricle pressure would increase, it should when it increases more than the aorta, it should drive flow through that valve. But in the case that this wasn't working, you could have some flow back into the ventricle, even if you're contracting. To get around that, they'd make these plastic structures, which is seen here. So this is the Hufnagel valve, and what it was is basically a ball valve. So the ball in its closed state would sit on this little hole here and block off any flow going through that hole. When the pressure in this segment of the aorta was lower than in this section here, then this ball would unseat from that hole and allow flow to kind of get through. When pressure was higher, it would close, and vice versa. So the problem with this design is a couple of things. So the first is that there's a lot of volume that goes in here, and a lot of stagnating volume. So that means like non-moving volume of blood. Whenever blood is not moving, it's gonna clot up. So it's gonna basically, like, when you cut yourself and your blood clots up, that's what happens in these structures. That's bad because those clots can dislodge and end up somewhere like in your kidneys or your brain, for instance, and that's not good. The other thing is that this ball slams into this hole when it opens and closes. That can squish some blood cells that are within this kind of orifice and also cause blood damage. So these were really, really, really bad. While like functionally they worked, they caused a lot of blood damage, and so these were no good. They transformed this into basically a device that was, instead of requiring you to cut open a blood vessel, a device where you could just place it with, much less invasive surgery. And again, this was still a ball cage valve, and you have a ball, and this time you have a cage, and it works in a similar kind of way. When the pressure in the atrium was bigger than the ventricle, the ball would unseat from the hole and cause flow to go around it. When the pressure difference in the ventricle would get much bigger than the atrium, like during systole, this ball would move back and basically close, not allowing any blood flow to go the opposite direction. The problem again is that you still have a slamming of the ball on the orifice, so that's going to cause blood damage on the outer edge of this seat here. So while it was easier to, I shouldn't say easier, while it was less invasive in the sense of, implanting this device, you still have problems of blood clotting and blood damage due to this. We've now entered a point where we're actually able to make a prosthetic valve that mimics the actual mechanism of heart valve. So there are no tendonae coordinae. So you'll basically place this 3-leaflet valve, which can be seen here, and there's no coordinate tendonae, but there is some mechanical rigidity to holding these valves open and closed. And so the way it works is when pressure increases from right atrium to right ventricle, um, this valve now opens up like flaps do and closes when the pressure gradient is reversed. Um, they were able to make basically get tissue from like a pig or some anatomically similar animal to us, and basically use that and just basically place that tissue on a graft, and they're able to place that. So in terms of the surgery, I'm not an expert at this. Obviously, I'm not a medical doctor, but depending on the valve, they can leave the non-functioning valve in there and just open it up and place this artificial valve in place of that, with the valve still, the non-functioning valve still there, just opened up. So there's all sorts of ways to do it, but that's the best way. What I didn't mention, where you have these other disc valves, so when the ball valve didn't work, they replaced the ball with the disc, and this did much better. But there were still some problems with the slamming of this disc, and so these are not really used anymore. Where they're using now these bioprosthetic valves here. What I will say is that, so what kind of engineers need to work on this? So this is all mechanical engineering, and within mechanical engineering, you have the mechanism to open and close a valve. You also have to consider reducing frictional forces with the contact of the valves to whatever structure they're attached to. Biomaterials is really important here, in the sense that you have to make all of these devices biocompatible, or at least inert to your immune system getting a hold of them. You also need in mechanical engineering, you need a fluid mechanics person, so being able to design these valves and then simulate such that how flow is going to be going around this structure here. That's really important. We'll get to that later when we talk about biomechanics. The flow profile of the blood going through different structures does matter, because if there's anything that causes stagnation in flow, that's going to cause blood clots and blood damage. So all sorts of things to consider, and this is still an active area of research that you'll find. So this is prosthetic valve. So I don't know, I think one group might have this, but just to give you a heads up that this is one area of research that is pretty interesting. Okay, another one is vascular stents. So stents are devices that open up an artery that's clogged. And so let's go back to this picture on the left here, or your left. Healthy arteries have very smooth inner walls, very similar to the endocardium in your heart. And sometimes, depending on certain diseases, it could be genetic, or just could be eating too much fatty foods, can cause plaque buildup in your arteries. So plaque buildup is basically fatty deposits on the inner lining of the vessels. And with that comes a constriction of the blood vessel, right? When you have constriction, that means that you need more pressure to generate flow through that artery, or that blood vessel. So one way to kind of to ease this problem is to open up the artery again, and so you need these devices called stents. So there's these metallic structures that are collapsible, and they're collapsible down to on the order of 1 millimeter or 2 millimeters in diameter, and that allows you to basically feed it into a catheter, which is a small diameter tube, where you can basically access blood vessels in your body. So, like for instance, they'll insert it in the femoral vein or femoral artery. In this case, the vein that's in your near your leg, near your groin. That tube gets then you can route it to whatever part of the body you want, because it's collapsed. It's easily you can easily navigate within circulation. And then once you get to the site where you have plaque, you can then use a balloon, which is underneath or within the stent, the collapsed stent, and that balloon will then expand the stent and push against the walls and the plaque, and open up the blood vessel again. So that's one way to do it. This is using a balloon to inflate and expand the stent. There are some stents that are just self-extendable, or expandable. And so what that means is they don't need a balloon to basically inflate them. So there's a sheath, basically another tube that covers the stent, and these are very elastic stents. So as you remove the sheath, the stent will then slowly start to expand, and it'll expand to the size that you need, and then the catheter is removed, and you leave the stent there, and your blood vessels opened up again. One of the problems with these devices is that, at least in the past, is that you do still have some plaque buildup, or you'll even have some endothelium, which is the cells that are within the inner lining of the blood vessel, kind of building up over the stent. So you'll have basically what's called restenosis. So it basically closes up again over time. But usually that doesn't happen for years after the surgery. So that's vascular stents. And again, in this application, mechanical engineers have to be able to design a stent and have basically a tube that's able to collapse and expand to very small diameters and then relatively large diameters, like 2 orders of magnitude bigger. So the materials also matter again as well, and the method by which you're basically placing this device is also important. One thing we I guess take for granted is that you do have a tube that's going in your vasculature, but that's going to have some resistance. So you want the doctor to, when he's placing the catheter in the bloodstream, for it to be not so resistive to him moving and maneuvering the device. So that also has to be taken into consideration. Okay, and again, I mentioned this even in the heart valve. So, in this case, you have mechanical engineers working on how stresses and strains are acting in this structure. Specifically, what's important is the radial force that you need to apply to open up plaque. You also have to figure out the fluid flow through the device, how it's implanted. There are some stents that are biodegradable, so you put them in, they open up the plaque, and then they eventually just dissolve into the blood. So that's also an interesting area of research. You can also coat some of these so that make them more biocompatible. So all sorts of things you can do with these devices that make them easier and better for patients that need them. Okay, so I've mentioned pressure quite a bit in this lecture, dealing with the circulation. So let's get to actual numbers here. So this is from here on out, with the exception of the nervous system and the ear and eye anatomy, we'll start getting into some numbers. Alright, so let's define some things. So I'm going to use the word hemodynamics here for a lot of the lecture. So basically that corresponds to the forces or mechanisms that are involved in moving blood in your circulation. The unit of pressure measurement, we all as engineers know that the unit is pascals or kilopascals or megapascals or even psi, pounds per square inch. But doctors like to use this unit called millimeters of mercury. And I've already gone over kind of the history behind that, but here's a little diagram to show you what that really means. Let's say you have a little glass tube. You fill it with some mercury in this case, and you have an unknown pressure going into a capped spout at the end of this U-shaped tube. You have atmospheric pressure in this open air tube here, so this is open to air. So atmospheric pressure is P naught. When that unknown pressure hits this interface between the gas and your mercury, it will basically lift this side of the U shape. So remember that when P is P naught, when they're equal, when they're both at atmospheric pressure, the height difference is 0, right? That's just due to gravity. And when you have an external pressure impinging on this, then you're going to have an increase in the height. And that height is what we measure in millimeters of mercury. So this is the way they used to read it. This is called manometer. This is a gauge pressure, so a pressure difference, and the reference pressure is atmospheric in this case. I say that because it's not 0 pressure, it's actually 760 millimeters of mercury. approximately, depending on the elevation. But that's atmospheric. So the pressure we read is going to be on top of or below 760 millimeters of mercury. Okay. If you want some conversions here, 1 millimeter mercury. So 1 millimeter increase in mercury within this tube corresponds to 133.32 pascals, or 0.01, basically 0.02 psi. Okay, so let's give some context to what this means. So when if you go to the doctor and they tell you what your blood pressure is, you're gonna have a number. They'll give you two numbers actually, and let's say that they told you, oh, your blood pressure is 115/75. So what does this mean? So this means that when they're measuring your pressure with this device that we'll get to later, that the highest number is called your systolic blood pressure. So that's the maximum pressure that your heart can generate in a contraction. So this is going to be 115 millimeters of mercury. That's 115 times 133, and that gives you the number of pascals. Diastolic pressure is your filling pressure. So that's like the pressure that's in your aorta or within your arteries when your heart is filling up. And so you'll see that it's not zero, and we'll get to that a little bit later. But again, it has to do with the fact that your arteries are expanding, and so this is the lowest pressure you'll read. So what does this mean in terms of your health? So again, I'm not an MD, but a lower systolic pressure means that your heart does not have to exert so much force to generate a certain blood flow through your body. That's good, because if you need 5 liters a minute of blood flow to your organs, if you only need 115 millimeters of mercury to do that, that's great. That means that your heart is working efficiently. If you need 160 millimeters of mercury to do that, that means that your heart is overworking and there's something wrong. And there's all sorts of things that can go along with that. It's not just the blood vessel itself. It could be with anxiety or stress, and we'll get to that again later with the nervous system. Diastolic pressure is a metric of how well your heart is filling up and also getting rid of that blood volume in your heart. The lower the number, the better. So what that means is that when you're filling up, you don't have as much volume left over in your ventricle. So that should be really low. A high number is like on the order of like 90 to 100. That means that your heart is still full. So you haven't ejected that fluid. So it's basically, think about this, is that the diastolic pressure should be lower because you want to fill the ventricle, right? If there's any fluid left in your ventricle, the pressure gradient from your atria to your ventricles is much smaller. So that means that you have less flow, but also that you're not emptying out your heart as much. So that's a concern, and it could be for multiple reasons. This is just to give you some context. 100 millimeters of mercury is about 2 psi, which is about 16 times less than the car pressure of your tires. So we're talking about pretty low pressures here in your body, and that makes sense. That's the reason why there's nothing exploding in your circulation. All right. So here's a little diagram to show you how the pressures change along the different blood vessels in your body. So let's say we're starting at the aorta. That's the blood vessel that's closest to your ventricle. Remember I told you that you have a lot of pulsatile flow through there? You'll have lots of peaks and valleys in there. And then as you get smaller and smaller from the aorta to smaller blood vessels, the dampening of that resistance starts to take effect, right? The actual magnitudes of the pressure fall off, but also the pulsations start to fall off. And once you get to the capillaries, you have no pulsatile kind of pressure in those capillaries. And you even start to have a really really drastic decrease in the actual pressures. And once you get to the veins, the venules, small veins, and large vessels, you almost have zero pressure. This is almost like atmospheric at this point. It's not exactly zero, but on the order of like 2 millimeters of mercury, 3 or 4, and then you start returning back to the right atrium here, and that's where you start getting, again, pulsations back because the right ventricle is then contracting. But notice how this is a drastic difference on the order of like 20 millimeters of mercury up and down. Here's 100. The right ventricle is much weaker in terms of generating pressure, and again, that's because the lungs are less resistant than like all your organs are. So then, but you have a similar kind of behavior as you go along in the blood vessels. They get smaller and smaller, the capillaries, and so this is where they turn into, they interface with the alveoli. The pulsations go away, and you have, again, small amounts of pressure here in the veins, and then you have a cycle, right? Once they go to the pulmonary veins, they return back to the left atria and left ventricle, and you get big pulsations that end up dying out and going in a kind of circle in this case. All right, here are a couple of different metrics, and this is not too important in terms of you memorizing anything like this. It's just to give you some definitions. Some doctors will use what's called pulse pressure. It's just the largest change in pressure during systole. So basically, it's just the systolic pressure minus the diastolic. That's the pulse pressure. The actual characteristics of these pressure waveforms does mean something, and we'll get to that a little bit later when we talk about bioinformatics and that kind of stuff, and trying to make sense of some of the things we measure. But we'll get to that later. Mean arterial pressure is the average blood pressure during a cardiac cycle. So on average, you're in systole for about a third of the time, and you're in diastole, which is filling, for about 2/3 of the time. So that's how they get this calculation where you can calculate the mean arterial pressure by P systolic times 2 times the diastolic pressure, all divided by 3. And again, that comes from the fact that you're in systolic for about a third of the time, and diastolic for about 2/3 of the time. What that means is, like, in 1 second, if you have 60 beats per minute, that you're in systole for about 0.3 seconds, and you're in diastole for about 0.6 seconds. Another metric is called cardiac output. This is the amount of fluid that the heart pumps in 1 minute. That's measured in liters per minute. It's kind of like current, right? It's just how much of something or some volume passes in a cross-sectional area over a certain amount of time. So that's called cardiac output. There's a thing called systemic vascular resistance. This is kind of basically like the resistance of your arteries lumped up into one resistance. Think of back to like 240 when you had a bunch of resistors in parallel and in series. You can combine them to one equivalent resistance. This is basically that metric. It's if you were to get all of your arteries and arterioles and capillaries, basically the load that your ventricle sees, and define it as one resistance value. And that has units of millimeters of mercury per minute over liters, which is just, think of like Ohm's law, right? It's basically pressure over cardiac output. And there's a relationship, which again is like Ohm's law, that tells you that the blood pressure is equal to cardiac output times systemic vascular resistance, where cardiac output is like current, blood pressure is like voltage, and SVR is like resistance. All right. Just to give you some context as well, a 70 kilogram adult male has about 5.5 liters of blood in their body. So your heart does a lot of work. If you have 5 liters a minute in flow coming out of your aorta, that means that you have almost the entirety of the blood pumped out in 1 minute by your heart. So it does quite a bit of work. Okay, now here are some more detailed definitions, and I'm running out of time here. I need to go to another meeting in a second, so we'll end on wherever we end. So one more or a couple more definitions. So end systolic volume. So now I know this is gonna seem a little bit annoying, but just think of how the words come out. So end means at the end of systole. So after your ventricles have contracted. So this is the volume after your ventricles have contracted and is done with the systolic cycle of your cardiac cycle. So like what you see in a schematic is like once your ventricles contract, they're not going to be emptied out. There's going to be some residual volume left in there, and that residual volume is going to be what's called the end systolic volume. We'll get to that a little bit later and what that means in terms of like a pressure volume loop. On the other end of the spectrum is called the end diastolic volume. So again, it's the amount of residual volume at the end of diastole. So once your ventricles have filled up, what is the volume after they've filled up? And what's also important is that end diastolic volume is also considered what's called preload. We'll get to that when we talk about muscles later on. But this is basically like, think of it like a spring, right? When you have a spring and it's uncompressed, right? It's just a spring. When you apply a force to it, it's gonna then start to compress, and you're basically like storing energy in that spring. This is kind of how the ventricles work as well. They're not springs per se, but when you compress them before you let them go, that's called preload, and that's the amount of load that you have before you contract. And that's a really important metric that doctors go off of when trying to diagnose if your filling dynamics in your heart are good or bad. Stroke volume is the volume that is ejected by the ventricles during systole. It's not the same as systolic volume. This is the residual volume once you've contracted. Stroke volume is the amount that is actually ejected from the ventricle. So stroke volume plus end systolic volume gives you the entire volume, theoretically, of the ventricle that's within the ventricle. Heart rate, that's just the number of contractions per minute, and that's in units of beats per minute. Here's a couple of equations. So stroke volume is equal to end diastolic volume, the residual volume at the end of filling, minus the end systolic volume, which is what you are left with once you contract. So that's how these are all connected. Cardiac output is just equal to the heart rate, so how fast you are contracting, times how much you're contracting in terms of volume, and that's again in liters per minute. Okay, I think I'm just going to quickly go over the beginning of this, and then I will continue in another video, so I can at least process this video for you guys, and move on to the next one. Alright, the cardiac cycle. So I'd mentioned this cycle here, and let's kind of get into the nitty-gritty about it. Alright, so I'm going to break up the cardiac cycle into a couple of different phases. So on the left here, you're going to see a heart and what's actually going on in the heart in terms of the valves and the flow of volume within the actual structure. In the middle here, we're going to go over this thing called a Wigner's diagram, and it sounds funny, but what it's going to do is it's going to map out the pressures in your ventricles, and in this case I'm going to focus on the left ventricle on the left side of the heart, but the actual waveforms look very similar, not the same. On the right side, just the numbers are a little bit different. So focusing on the left side, this will show you the pressures in the ventricle, the pressures in the atrium, and even the pressures in the aorta in this case. So the 3 different pressures, and you're going to see how the pressure gradients are changing. On the bottom part of this graph, you're going to see the volume within the left ventricle. So how is that changing in time? And this is the x-axis is basically time. So in this case, we're going to assume that this happens over 800 milliseconds. So let's go over it. So the first phase that we're going to go over is called, and again, it's no particular order, but this is ventricular filling, or also called diastole. So let's start off there. So in terms of what's happening in your heart, that means the pressure in your left atrium is higher than in your left ventricle, but the pressure in the aorta is also bigger still than in the left ventricle. So to map that out on a plot, that's seen right here. It's where, so the dotted line on the bottom here is the left atrial pressure, the red line is the ventricle pressure, and the dotted line on top is the aortic pressure. So notice the kind of the directions here. The red line, which is the ventricle pressure, is below the atrial pressure. That causes movement into the ventricle to fill it up. But that red line is also below the aortic pressure, so that's here. And so again, that prevents flow from moving from aorta to ventricle. So you only have one-way filling from the left atrium. That can be seen here if you're looking at the volume. You're starting at a certain volume and then you're basically increasing a little bit, and that's where you end here. Now, here, where you end, when you're done filling, that is the end of diastole, right? So that is called the end diastolic volume. And LV stands for left ventricular end diastolic volume. Right, so that's just the preload of the left ventricle. So that is diastole in a nutshell. Now, another way you can view this is through what's called a pressure volume loop. So this is the same plot as this. Now, instead of time, instead of looking at pressure and volume in time, you're looking at pressure and volume together. And time is within this plot, but you can't see it explicitly. So if you plot the volume change versus the pressure, the ventricle pressure is the red line. So you're going to plot the red line and the black volume line down here together. And what you're going to get is that you have a change in volume, right? Now these numbers don't match up over here, just for the record. This is a different plot than the one from here, but it's the same concept. So like say here you start off at, I don't know, it looks like you're starting off like at 100. But, so you start off there, and what that's basically saying is you're starting off somewhere here, and you're increasing to 120, so that's about here, 120. And so you're going along, so volume is changing quite a bit, but the pressure is not changing much. So you'll see that you do increase a little bit from, something above 0 to something a little bit closer to 20, but still not really much of a change. So in this case, you just have filling, but you're not having much of a pressure change. And this is just mapping out the different pressure differences. And let's just go over one thing and then call it a video here. So that is diastole. What I should have mentioned, that this is the end of diastole. Diastole also is over here. So this is like late diastole. This is early diastole. The reason why I pointed here is because say you're starting off at like 90, you're like right here somewhere, and increasing a little bit. The whole entire diastole period is like from here all the way through this entire section here. So you'll have from A to C goes from 425 milliseconds all the way to 100 milliseconds over here. Anyways, so we'll get to that a little bit later. So this is the end of diastole. Once you get to the end of diastole, you get to systole, which is called isometric contraction. So that's right here. It's right before you eject anything. So for a very short period, you're going to have an increase in left ventricular pressure, but that is still it's bigger than the atrial pressure, so left atrium, but it's still below the aortic pressure. So nothing is actually coming out of the ventricle. It's just that this is now like contracting and building up pressure. If you look over here, the red line is now above the dotted line, which is, again, you have no flow from atrium to ventricle now because the valve is closed. But it's still below the dotted line on top, which is the atrial pressure, so you have no flow going out. So that's why it's called isovolumetric contraction, because you're increasing pressure and your ventricle is contracting, but there's no volume change because nothing has been ejected out. And that's seen right here, where you see like there's no change in volume here. On the pressure volume loop, that's seen as just an increase, basically a vertical line on the pressure volume loop. So that just goes up. So you have an increase in pressure with no change in volume here. So that's this leg of the plot. Now, once this pressure builds up such that it's greater than the aortic pressure, you'll start seeing some movement of volume. And so we'll go over that next time. Like I said, I'm running out of time. I have another meeting. I will process this now and then post it So at the very least, you should have it for today. And then I'll try to get the other section of this recorded and then up by Tuesday at the latest. All right, thank you, and have a good day.